Yo everyone, welcome back. Herb's Happy Hour, Friday, 5 p.m. Pacific Standard Time. Thank you for joining. And if you missed us last week, we had Bill Burr on here. Super funny. Such a good guy, man, and a really great drummer. We did some drums, talked about drumming, talked about music. He's always on. He's amazing. So thank you, Bill. If you happen to ever come back on this channel ever again, thank you for coming on and getting in touch with people and letting them know what's up behind the kit. Yeah, so you can also find that. Hold on, guys. Wait, I might need a little bit of my happy hour drink. I am having I know it's backwards. It's backwards. Herb cider wine sap mezzo forte. This is a wine sap apple. It's one of my favorites. You guys can get that on our website too. You know, join the clubs too, and maybe we'll have some of that going out again. Anyways, let me get a drink. So yeah, fortunately, we've been able to save these videos, and we have Instagram missed our first 3 because they weren't allowing it to save. My door is squeaking. But we have them saved on our Instagram channel right here on the Herb Cider Instagram, and you can also see them on the YouTube channel that we've created, the Herb Cider YouTube channel. And there you can subscribe, you can like us, you can do all that cool stuff that makes our channel the best. Better than, who knows, better than everybody. We need billions of views. So tell your friend, come check it out. If they want to see some silly hangout, talk about cider and music and all kinds of stuff, you know. Which, before we bring on our guest, we are having a guest today. It's not just me. And can I tell you, I've just been thinking about it. Whenever I start these dang things, it's quiet in here. I'm by myself. I'm just sitting here. And then I hit the button that says live. And then, oh, hi, Charles. I see you there. Charles is going to come on. Give me just a minute. Yeah, it's the weirdest thing. It's dead quiet, and then I just have to start talking to no one about whatever. So I don't know. You guys try it. Sit at home and then just start talking to yourself while you're looking at yourself in a camera and trying to entertain or do something. It's the weirdest thing, I got to tell you. Anyways, I just wanted to tell you that in case I look silly up here trying to figure out what I'm doing, but that's why. It's so weird. All right. And yeah, we've got my wine sap you can get. And also, you guys, check this out. This is the new Birdie Juice can that will be available this week, I think. Are we having it next week in stores? But we're going to be canning it this next week, and it's pretty cool. There's a little story to this too. So see that guy right there? He is our Primus video guy, Agent O. So we were what we do basically when we're on tour, we get days off, and some of us like to go play some golf, hit the ball around. And this was a special time where we were on tour with Mastodon, and every day off, and then some, even on show days sometimes if we had time, we go play golf. And it was me, it was Agent O, my video guy, and then from Mastodon, the drum tech, Norm, and the sound guy. We would all get together and go play these courses, and they were the coolest guys. So 2 guys from each band, we'd go out and we'd hit. And Rob was his name, sound guy, Norm, Agent O, and me. We go play some golf. But this time right here, the Mastodon bus and the crew bus for Primus, they all stopped off at Pebble Beach. Primus bus was 2 hours south, down having a day off on some, I forget what city it was, on the beach, San Bernardino or something. And I'm like, God, man, I've never played. I gotta go. So I rented a car. I drove 2 hours north. I get there, and Pebble Beach had already set up the foursome of those 3 guys and a single to make 4. And so when I got there, I had to sweet talk the guy who was with my guys and say, hey man, you mind if I jump in? And our time was at the very last tee time, where when you get to 18, the sun is going to be setting. So there's no guarantee that you'll finish 18. So we managed to finish 18. So this is a shot from that day, and it is on the Birdie Juice can. So it's pretty cool. Check it out when we get it out next week. Come online, herbcider.com, and you can check that stuff out. What's up?
I saw Charles hop on. I think he's probably ready. Do you want
Yeah,
to
I told Charles I said I would get to him in just a second. I just wanted to tell everyone about this. So yeah, let me. So today, you guys, we're bringing Charles on. So he is our master scientist, virologist, and we had such a good conversation, and everyone was saying how cool it was to have him on. There he is. Hey, Charles.
Hey.
So yeah, everybody was saying last time that they were so informed by everything you were saying.
It was the most informative.
It was the most informative talk that people have found out about COVID and the coronavirus. So, you know, that's why I was thinking, man, let's get you on again, because when we talked, you had tons of questions. I mean, we had tons of questions about what we were talking about.
Yeah.
And we had to end. You know, this thing ends at 1 hour. So
yeah,
thank you for coming on and doing this. You know, doing it
it's
again.
my pleasure.
And
yeah,
you said
totally
you
cool.
did some research for us, right?
I did a bunch of research. I've got lots of charts and graphs.
No.
Yeah, we can
Graphs?
and yeah, and we could talk about that, and we could revisit a couple things we talked about last time too. Even
we
the
need
masks.
to, for sure.
Yeah, like catch everybody up, do a recap, and go with it. I'll follow your lead.
All right, hold on one sec. So everybody out there, you know, if you're watching, you can put a question up. Shama's here, and she's watching the feed, and she'll yell at me and tell me, hey, you know, this person asked this question. So
Awesome.
yeah, we'll try to monitor the questions and keep it moving.
Cool.
You know, so we can get some info on this. You know, yeah, well, you want to go back to kind of reference some of that stuff we talked about, because I know, it's still a weird, confusing thing, you know?
Yeah, it's totally confusing. Let's start with the terminology. I think that's really crucial.
Yeah, okay.
So there are 2 different things, and even it gets mixed up all the time, and I see this all the time. So there's multiple things that mean different things, and it all starts out with, what's the virus? So we know there's a virus. It's called a coronavirus, and that's a family of viruses. So there's a whole bunch of different types of viruses in that family, and the one that we're dealing with right now has a funky name that people don't even use in the media, and it's called SARS-CoV-2. And that's the name of the virus.
That's the name of the virus, yes.
That's correct, yeah. And then we just refer to that as coronavirus, and that just makes it simple. It's kind of like saying I got the flu, but in fact we got a specific kind of flu, like H1N1 or whatever.
All right.
So that's that. And then that's not the most worrisome thing. So the most worrisome thing is the thing called COVID. It's called COVID-19, and COVID stands for, I think I've already forgotten. I don't know, but that is the disease caused by the virus. So we get infection with SARS-CoV-2, also known as coronavirus, and then that causes this crazy respiratory distress, and then that disease, that respiratory distress, and all the associated issues with it is called COVID-19.
Wow.
So that's the distinction.
Okay. What were you saying?
People can kind of think of it as like HIV and AIDS kind of thing, right?
That's correct. That's correct. So HIV is the virus, and then AIDS is the results of the infection from that virus. That's absolutely correct.
Right. Yeah. So similar thing going on here.
Yeah.
And is it? Do you know? I mean, if you get the coronavirus, is it COVID that you get, or can it trigger other things?
Yeah, so there's that's a great question too. So the first thing is that a lot of people get COVID, like somewhere between 96 to 98%, I think. Maybe it's not that high. Actually, it's not that high. But a majority of people that get exposed to COVID get either they're asymptomatic, which is really worrisome because they can spread it. They get a mild disease, so not even disease. They get some flu-like symptoms, body aches, headaches,
Right.
some really dry, pronounced cough, and those people are okay. And then the for a small percentage, I don't know, I'd have to look at the exact number. Is it 10%? Is it 20%? Then they get the respiratory distress, and then if it can get very serious, and that is age dependent, it's the health dependent, and I've got a chart on that, and that's when the disease can actually get more dangerous. So if we get exposed, there's no guarantee that we, as people in our 40s or 30s or 20s, especially the kids, as you get younger, the likelihood that you'll develop any symptoms at all is reduced, and then we're generally healthy people, and our immune systems can handle this, and we may get just mild symptoms for most people.
So is that something that's being, those kind of figures you've come up with, actual like data from this for the last, whatever, it's been like 6 months probably since it's floating around, that
so,
the age thing does matter?
Yeah, for sure. So
Okay.
what a good scientist does is, you know who they are, you know where they studied, and then they're willing to show you their data and then tell you where they got their data so that you can follow up on it and look into it yourself. So a friend of mine from Sacramento found this awesome website, and I haven't figured out who these people are yet, but the data site looks really convincing. So I presume these people know who they are. I need to look into it.
It looks
but it's
convincing.
It's totally convincing, and it's consistent with what I've seen too. But it's a nice, I think what they're doing, it's called Our World in Data. I'll show you that in a second. ourworldindata.org.
Okay.
And they've, like, all these things that I've been hearing about for 6 months, I never see them in one spot, and they're putting it all in one spot. So they're aggregating, I think, the data worldwide from all kinds of sources all over the world and putting it in one spot. And if I can get this up here, is that backwards? It looks backwards, right? But there's
it is backwards, so we can't really read much about it, but you
yeah.
can tell us.
Yeah, so at the bottom is this is basically age. So at the bottom, these are the people that are most affected, and that's probably 70 or to 80-year-old, and then up here are the kids, and so they have very few effects. Yeah, you can see it. I think it's grouped by 10 years. The older you get, the more likely you are to have major problems.
Yeah, this is the thing that people kind of don't put together, is what I'm guessing, is when you see people out there, even the kids, which I wanted to ask you about with the school and stuff, but
yeah,
you got the kid goes to school, comes back and sees grandma and grandpa. I mean, you theoretically have a possibility, if that kid had it, of killing your grandparents.
That's exactly the concern.
People don't put that together. We're having a lot of pressure here with our school, and
yeah.
They're reopening.
Anyways, that age thing is really important.
They're reopening in person, Charles.
Yeah, our school is reopening with kids in the classrooms. They're doing all the like masks and glass boxes and stuff, and it's like, that's going to be horrible. We have the technology to sit at home. I don't know why we just can't manage at home. I know a lot of people got to work, so school is almost like babysitting, in a sense.
Yeah. We
So
need it. Yeah, it's a really hard
it's
decision. I
tough.
struggle with it too. And even my wife and I are on, I wouldn't say we're on different ends, but my wife is like, no, we want to do online learning and we want to, we want to do all that. And I'm like, no, the kids got to go back to school. It's like, because we gotta work. And
Yeah.
so our district decided to do wholly online. And I got 4 young kids, third grade,
Wow.
fifth grade, and 2 seventh graders. And and it's working. The kids get distracted. You can imagine all the problems, but
Right.
I think that's the right thing to do right now because there's this risk, and as you say, putting it together, the kids and the grandparents, right? So what isn't understood, that's the other thing a good scientist does, is they tell you what they don't understand yet. And so what's not understood yet is, can that actually happen? So if you have a child that has the virus, coronavirus, and I saw Fauci or somebody was looking at the viral titers. It's the amount of virus that you can swab out of a nose and do PCR testing. So that's high. It's like as high as people with symptoms, but they're asymptomatic. And so the question is, can a child that has the virus, can they actually transmit it? Is not the known. It's assumed that can happen, that the transmission can
Oh,
happen
it's
to
just
someone
assumed.
else. Yeah, it's assumed right now, and I
Ah,
think it's probably likely, but the data hasn't come out yet where we know that if we, put a bunch of kids in a room, that they will actually transmit it to each other, because kids somehow have this, protection against all of this that nobody understands yet. It's not complete protection. I saw one report where like 200 kids in Florida died, of COVID. But that's far
dang.
less than the 8,000, other people that have died. So, but it's still, so there's still some risk, but nobody understands yet the transmission for children, and then when the children are super susceptible and there could be potential mortality. So, but I think it's safe. The thing we need to do now, we can't wait for science to catch up. This is good. These studies are going to take, after you get the data, it takes 2 or 3 months to write the paper, and we can't wait for that. So I think the
Yeah.
right response right now is caution, which is either keep your kids at home and homeschool, or it sounds like your school is taking precautions, which is what they can do. So that's also good.
Wow. Wait a minute. I saw someone here that says my school has us wear masks and we sit 6 feet apart. I mean, what do you think about that? Is that really effective? I mean, we've talked last time about masks.
Yeah.
That was one of our questions. Can you go? Did you find anything about that? Like
Yeah.
the different
So
varieties, kinds of masks?
yeah, I found a lot about that. So there's a study. So where I was masks, recapping, right? So masks will help reduce exposure, and distance will help reduce exposure. And then if you have no exposure, which is quarantine or some bubble where you feel safe with a limited number of people, those are all good things. And then with respect to masks, it turns out it's not so black and white. Like if you wear, I actually have some props here. So here's a surgical mask. So this one you put on over your
Oh,
nose, you
right,
tie it behind your head, and it's got this wire in it which can seal around your nose, which is
right.
important. And then there's this other style here, which is it's got the ear loops like this, which is also good. So these are pretty good. They're very good. And then there's the N95s, which are even better, which I don't have an example of.
Right,
And
right.
and so those are all good, but they're not perfect. And I'll give you some stats in shortly. And then there's this thing, which is which you basically put it over your head, right? And
Oh,
then you
yeah,
can
yeah.
just pull it up like this, right? When
Right.
you see people. This one actually is believed to be not so effective, and it might even be worse because it has these pores, and if you cough, it can actually split up the droplets and make more droplets, which
Oh,
is not
whoa,
obvious.
really?
Yeah, yeah. So it sounds like either N95 or some kind of
So hold
surgical
on.
type mask. Yeah,
In So what you're saying, if a droplet gets
yeah,
split up, does that mean that the virus there's like what? How many I mean, how many viruses or viri
yeah,
is there in droplets as they come out? Is it because I guess like an idiot, I was just thinking, oh, there's a virus in a droplet. How
yeah.
many how many
It's
can
a
be in a
good
droplet?
Yeah, it's a good question. I don't fully know the answer,
Oh,
but let's
okay.
say someone has it'll most likely be multiple viruses per droplet, and then there's all this. I was reading the papers; it gets really crazy because then the size of a droplet has it's got a certain volume, and then how many viruses can you put in that, and it gets really complicated.
Wow.
But the idea is that there would be definitely multiple viruses in a single droplet, and then
Yes.
when you split that, then you just magnify the kind of the spore. They're not spores, but that's the idea. Like
So we're
you would put
probably
more of this.
talking thousands, right? They're
Yeah,
microscopic, right?
Yeah, thousands or ten, ten
Tens of
thousands or like,
thousands.
yeah, 100. It could be hundreds. I'd have to look into those numbers, but
But we're
yeah.
we're
So
not talking about that level of mass
it's a
in the
lot.
in those droplets, right?
Yeah,
So yeah, so just so people know, the interesting thing, like you said last time, was a mask does not stop the virus, but it stops the droplet that the virus is in, right?
yeah, that's
Does
right.
that mean the
Yeah.
virus can only travel in the droplets, or what about what is it, the aerosol effect?
Yeah.
Yeah,
CDC came out with
how does that work?
more
So
information.
there's some like definitions here on size. So like so aerosol, and I'd have to look up these definitions too. But
Right.
basically, a droplet, somewhere between a droplet is a bigger thing, and then an aerosol is a smaller thing. And then a droplet would be something you could easily see. Like when we talk about sneezing or something like that, right?
Oh, I see. Okay,
It's
yeah.
like, oh, it's wet. You can see it, right? And
You
you
can
can
see it,
if
yeah.
it gets sneezed on, right? It's awful, right? And then
Right.
there's the aerosols. So what I didn't know is until I started studying all this is that as we talk, like we don't feel any droplets, right? Like you don't feel anything on your hand, but we're
Right.
actually emitting aerosols. So those are just tiny, invisible. It's a tiny droplet is essentially what it is that you can't see, and but has a volume and a small size. It's microscopic and you can't see it. And then those are what are they? If you can't see them, they're below a millimeter. So like a 0.1, like a 1/10 of a millimeter or less is probably what we're talking about here. And then those can kind of float around and they can stick around for a while and they can hang out. So
How long a
that's the
while? What do you
distinction.
mean?
They're talking about this a lot. So there's it's not as black and white as the 6 feet even. When I was first learning, like the first models were like, oh, if you're 6 feet apart, then the droplets will, and then I think they're talking about bigger droplets, will go out and then they'll fall away, within a few feet and they'll drop to the ground. And then even if some kind of travel further than 6 feet, you've got you're both wearing masks and then the second person will be affected. But it's not as straightforward as that. What I've been reading more recently, and I don't know if I have the references here, but those aerosols can kind of hang around and linger for a significant amount of time. And so that sounds scary because what I'm describing is a scenario where if you're in a room with someone and you're both wearing masks, that you're going to get exposed, right? And but I don't think we need to be alarmed about that right now because again, it's all about exposure and how much. So in that scenario where if you're outdoors, it's kind of like dilution. Like if you take a drop of herb cider and drop it into a glass, you probably won't notice it, right? And if you drop it into a pool, you definitely won't notice it, right? So it's this notion of dilution. So that's why outdoors is better because you have those things. If they're there and somebody has it and they're 6 feet away from you, it's going to go out and kind of dissipate into the giant volume of air around you, and you're less likely to get exposed. Now, the bigger risk is, and this is more related to indoor activities, like including the kids. So if someone's there can be some aerosols that get around that will be there, and but then again, when we have this mask on, then we should effectively reduce most or all of that. So I can't give you like a really solid black and white, like if you're 6 feet
Right.
apart, you're both wearing these masks and and you're in a room, a 12 by 12 room, that you're going to be fine, But
Right.
the thinking is that we're going to be okay. But there's just more to learn. And the only way we're going to learn is by either we stay at home for a while and we get that data, or like your school system's doing, people are going to learn from that and around the country. I know my friends in Illinois, somebody I went to elementary school with is a teacher, and she's going back to school. And so part of that is, we'll learn from that and find out what the
Right.
risks are.
Wow.
Yeah.
Yeah, there is a lot of vagueness to it all, and
Yeah.
that's what's hard. But yeah, I just definitely think having your kids sitting in a classroom is just maybe not a good time right now, because like you still don't know the stuff. Even
Exactly.
you who has some avenues of researching, you
Yeah.
still don't have all the data on what to do and what not to do.
We don't have all the answers right now.
Yeah, because if they're in a classroom, it can float over the plastic into the
That's
next kid.
correct.
It's like,
Yeah.
yeah. So we're dealing with that. I think we're going for online schooling for a while until this stuff gets clearer, you know.
Yeah.
What's
Do you
up?
Makes
give
sense.
feedback on the vaccines that are coming out right now, and like what
Yeah.
what you
Yeah.
know? There's two different universities I think that came out. I think it was Oxford, and then
Yeah.
Wazoo came out with one like a day later. Could you talk a little bit more about like
Absolutely.
Did you guys hear that?
and like the why they're so, you know, they're developing different vaccines in different places rather than working cooperatively?
Yeah, you heard all
Yeah,
that.
I can. Yeah, I can address all that. So,
Okay.
yeah, I looked into that. I figured that might be a topic, and so I looked into that last week and looked into this. And so there's I don't have an exhaustive list of everything that's going on, but I can tell you one of the reasons why there's so many of these and they're not cooperating is everybody's got their slightly different take on this, and I'll explain a few of those. And that's actually good. So that's the scientific market and the business working for us. So a bunch of different people want to be first, and they want to have the technology because they, you know, it's a business, it's capitalism, right? So there is that aspect. And then what I hear is the governments are buying these vaccines. They're poised to buy these things so that they, you know, the cost is not prohibitive and we can protect everybody, right? So I think that this is going to be a cool blend of capitalism, if that even plays a role, and safety for humanity. I mean, that's a nice thing that's happening. So why are there so many? So there's there's a couple ways to do this. There's actually 3 or maybe 4 that I can think of. So the old way, you know, the early vaccines, I think it was Jonas Salk, right, and polio vaccine, right? So he was able to make polio, actually grow the polio virus, and then he just heated the thing, and then it dies, and then he would take that dead virus and inject it into people, and then they would survive. And he figured that out before anybody knew anything about cell biology or virology or anything. He just knew that he had this, you know, this, maybe it was even a substance. I don't even know if he knew what the virus was, but he knew if he injected it that people would not develop polio, and so that that idea basically is you take the virus, and the virus has these it's little molecular machines called proteins, and it's the same protein as in like we have a hamburger, but these are very they're high architecture. It's folding, and they bind to other things, and there's sequences, and everything we're doing right now is related to proteins, like me seeing you, like me talking to you, my brain, everything. And so in a virus, those proteins are unique to that virus, and then our bodies have this amazing ability to distinguish between a foreign invader and self. And so when you inject the whole virus, the body says, "Oh, that's a foreign entity. I got to clear that thing," and it generates what are called T cells, which are immune cells, and then ultimately you generate antibodies, which circulate in your blood and then grab that thing. So that's the first kind of vaccine, which is basically an inactivated virus. The second thing that works better is where you say, okay, well, it's this one protein, and there's a lot of talk about the spike protein, which gives it that's the corona name. So you have the circle, and you have all these spikes off of it, right? And that's the spike protein is the first thing that the immune system sees because it's on the exterior. So what people are doing is just making that protein. It's called recombinant protein. It's you make it outside of the virus, and then you make a bunch of that protein, and then you just inject that into the bloodstream, and then again the host says, "Oh, that's not right," and makes antibodies against that. And then there's 2 more types. I know this is getting a little technical, and I'll stop shortly. And there's 2 more types. One is where you put that same protein in another virus called adenovirus. And adenovirus is something we all have and we're all exposed to. And then we would take that adenovirus. I don't know if it's injected or we inhale it or what we do. And then that adenovirus makes that protein, and then you get it that way too. And then the last thing is Moderna. Like what they're doing is I think I had it here a little while ago. Let's see, Novavax and Moderna. How are they doing it? They're doing one. I can't read my notes here and figure out which one it is, but they're doing one of these. And the last thing that people are doing is they actually take the gene. So the gene is a sequence of DNA, and then that encodes for the protein. And what people have figured out is you can inject the gene, like they probably do it in the muscle injection maybe, and then the body will just make the protein automatically. So those are all the ways that are going on. And if this can be done, and that's a big question. There's no guarantee that these vaccines will work. That's an important thing to know. But if it can be done, I guarantee you someone's going to figure it out between one of those 4 or 5 ways that I just described.
Right. Gosh dang,
I
man.
have a question about the proteins. So I know, like in
Yeah.
some sort of biologics, like Humira and Remicade, they use bovine proteins and rat proteins. Do you
Yeah.
have any insight if they're using animal proteins? Because I know that those can cause quite a
Oh,
bit
yeah,
of
yeah.
reaction.
Yeah, so people can have allergies to those things, I think is the concern, and maybe reactions. Yeah, and there are some things. There's one called IVIG, which is intravenous immunoglobulin, which is like from horse, and that's used for like horses get exposed to a lot of similar viruses, and then they make their own antibodies, and then some of those horse antibodies will actually cross-react with things that are threatening to us. And then people are actually using that for treatment of coronavirus. So somebody figured out that if you had somebody that was exposed to coronavirus, they could take their serum and they could purify their serum and give it back to a patient who's not who has COVID, and then those antibodies might help neutralize the virus. So that idea makes sense. And so sometimes it's real obvious. It will be from, they'll say, oh, this is horse, and that should be explained to people. I haven't heard anything about rat or anything like that. There are these things called adjuvants, which are basically, if you have that protein, it would be a human. Well, it's a viral spike protein, but they're generally made in various expression systems, and then that's where the FDA gets involved. And this is what's great about government is we have the FDA who says, okay, you got to prove to me that you've purified all that stuff away that can be a problem, or you got to tell me about it when you make it so that people know about it. So those things, if they're there, they should be known and disclosed, and they should not be a risk, is my thinking. Unless it's known that it's there, and someone's sensitive to those things, and then people can know that. So the purification processes are pretty amazing, and then the impurity profiles are known. That ought to be readily available to people. I'm less familiar with the clinical side and the actual therapeutic entity side, but that information should be there.
What do you think about, if they do come out with one, like, should people just go ahead and get on it and do it, even though there hasn't been a lot of years and years and years of testing?
Yeah, my thinking is this is going to be most likely. So vaccines are pretty well understood that the RNA, and I saw someone mention mRNA, which is correct. So those are, I know less about. If you inject that gene, and how does the gene stick around? Where does it go? How long does it last? I don't know much about that particular one. But if it comes from the protein or the adenovirus, those are pretty well known, I think. And the protein for sure is like that idea is really well known. People do that all the time. I think the flu vaccines, those are actually grown, and so those are inactivated viruses. So there's different comfort levels that I personally have with that. So I think we're going to have to, the question is, do we do it right when it comes out? And there's going to be one or two vaccines, maybe three. It's not like we're going to have a choice of 100 vaccines and we're going to have to go, which one do I take? This should be really, really obvious. And then if it gets out there and if it's available, it will work, and for some population, right? And then it's good. The questions you're asking are great. Then we got to say, is this the right thing for me? And I thought a lot about that question. It's like, do we just all go for it? And like, Putin saying he gave the Russian vaccine to his daughters already, and they generated antibodies and all this. Yeah, it's like crazy. They went out of, so there's multiple phases. There's phase 1, which is a small safety trial. Then there's phase 2, which is maybe a few hundred patients. And then you go to like thousands and thousands, 10,000 patients. And they skipped all that. And they just went, yeah, we got a vaccine, and we're calling it Sputnik, So for me, it's like, would I take that, or would I give it to my kids? It's like, hey, did I already have COVID? I mean, not COVID. I didn't have COVID, but was I already exposed? I had some wacky thing in February where I'm going, oh that was crazy, man. And then all of a sudden I'm like, wow, that was kind of similar. And my doctor said, nah, it's a flu going around. And I don't, but I'm like, how do you know? There's no testing, So and that's a whole other thing. We should test and understand. We might, if we've been exposed, we might have some immunity already.
Tell us about that real quick. We wanted to ask about testing.
Yeah.
We've heard a lot about what is it? They're not accurate. Is that?
Well, there's so many different types.
There's so many different types.
Yeah.
There's blood types. There's fecal. There's nose swabs.
That's the antibody test. So those are. And
so
then
what do you know? Like, what's the best?
curious about the efficacy of all these various tests
Yeah.
at different universities and labs.
Yeah, so I can comment on that, and what I don't know. So here's the problem. You've identified the problem. We should know which test we should be doing. We
Right.
should,
And
so we
everyone
should,
should
we
have
shouldn't be
it.
going to some Frappuccino stand that was, converted Frappuccino stand to get a COVID test.
Right.
It's like so the best way to go through is your doctor for sure. And or call them and ask them to recommend some place. The way testing works is, and this is my first job was actually in testing, and there's two types of tests to think about. One is do I currently have the virus or not? That's the first question. And the second question, and that's called a PCR test. So PCR is like, it was invented in Emeryville. What's his name? Kerry Mullis, Nobel Prize winner, wacky guy. So that really cool test. Basically, what you do is amplify the gene from the virus, and then that's a super sensitive and super specific test. Specific means if you do this test, you will get an answer, and it will be a correct answer. And so that test is good if if we want to go visit our grandmother, or if we're worried about our child, or these kind of things, and then you get that test. And but the problem is so that test is probably okay in general no matter where you get it, but the way the FDA works is that these tests get developed, and it takes 6 months to a year to develop these tests. And then you go to the FDA and you do all this. You put a package together. The FDA says yes or no, and then that test is approved by somebody who knows what they're doing. Like Roche is big in DNA type and RNA type testing, and so then that test would be ordered through your doctor basically, and then it's legit. There's no question about it. If you're going out on the to the mall or to the corner, whatever, then I just don't know. And the point that you raise is a really good one. We don't know where those tests are coming from and how they're being done. And so if you get a negative answer, what does it mean? Or if you get a positive answer, what does it mean? These are the concerns. So we nationally, we need recommendations on which test to use and where to get it. And I think ideally we would just know that, but the best thing to do would be go through your doctor, because they will know who to get the test from. So that's the virus test. And then the other one is the antibody test. So that one, like I just saw in Sturgis in North Dakota, right? There's the motorcycle rally there, right? And then they didn't want to wear masks and all this, and I'm like, oh man, yeah, that's what I'm thinking. And so there was a report in the local newspaper that oh yeah, they did antibody testing on everybody and everything's fine. Not everybody, but then some people. And so that's not true, because the antibody test doesn't tell you if you have the virus or not. It tells you if you made antibodies to it, and that means you were exposed at least 14 days ago and probably recovered from it, because it takes after exposure, and actually after a fever, then your body goes through a really elaborate process to make antibodies, and the first signature shows up at 10 days, and then the real signature that you measure shows up at 14 days. So at that point, they're probably not even infectious anymore, and they would know if they had symptoms. But it doesn't mean that they are exposed or not exposed, and it doesn't mean necessarily that they don't currently have the virus either. So
Really?
so that. Yeah, so because the presence of the antibody indicates there's false positives there too. So the antibody test is probably not quite as reliable. It's going to be a good test, but it's not perfect either. So the antibody test could be cross-reactivity with some other related virus, because that stuff takes time to sort out and to develop that test. And that's actually a business that I did work in. You got to look at all kinds of samples from all different kinds of viruses that might cross-read or give you a false answer on that one. So that's the trick with the antibody testing. But I mean, it is, I think the point, the bewilderment that you're expressing is that if you did show a positive antibody response, then you might say, okay, well, I probably don't have it. And if you don't have a fever, you're most likely in good shape too. And so if you don't have a fever and you've got antibodies, well, then you can say, well, maybe I'm doing okay, would be the and I'm at lower risk, and maybe I already had it, and I'm going to be okay for a while. But that's an assumption too. So now that we have the test, let's say we have the right test, we still don't know if you get exposed to it again in 3 months or 6 months, or that you're protected. So that's the study that study hasn't been done either. I think it's probably a safe bet, but the data's not there yet.
I had read that it was about a 30 to 40% false negative rate from Wuhan to the U.S., from when it started in Wuhan till now.
Yeah.
Is that true?
That It could be that high. I didn't see that particular report, but it would make sense. So
Yeah.
PCR is tricky too. So it's super sensitive, but you really got to have the virus, and it's got to be in your nose, you know. And so there's kind of like it's called there's a number for this where is you're basically like if you take samples like take a nasal swab for someone that's been exposed, like let's say they get exposed today, and you take a nasal swab, you won't be able to see it in the PCR test. You do the same test the next day, you may or may not be able to see it. It basically takes a few days where the viral titer gets high enough and gets into the nose until you can see it. So there will be people that have been exposed and maybe they haven't developed symptoms yet or haven't developed a fever where they will test negative, but they actually have the virus and they will develop symptoms and a higher viral titer and a positive test a few days later. So that definitely can occur.
So if you go get a test,
yeah,
like we've got a buddy who played golf in the U.S. Amateur just this week, and before they could even go into the course, you know, they all got tested right there, and they had the results, you know, almost immediately.
Yeah,
I mean,
so I think that's
is that legit?
I think that's as good as we can do right now. You know, I think there's a couple things that I think are really cool to think about. So sports is those guys are doing exactly what we need to do. They're using all the tools that they can use to make the best decision they can make, right? And so I think that is awesome. And so the golf thing sounds like the NBA. The NBA basically is like, okay, your record was good enough. We're going to Orlando, and your people are, you know, your team's staying here, and you can't leave for the whole duration of these playoffs. And your other team's over here, and you can't cross-pollinate, you know, and play foosball in between, or go golfing or whatever, you know. And you can't eat dinner together, and your relatives can't even, your family can't even come over, you know. So they're doing total bubble, and then they just go play, and so it's a highly controlled bubble is what I call it. And so you know who's got it and who doesn't, and then if someone develops it, then they're going to have a problem, and they're going to have to shut some parts of it down. And that's what Major League Baseball didn't do that. So they're doing probably some testing, but they're not controlling the environment. They're not controlling the bubble, and then they're having trouble. So like they're getting a couple of players, 3 to 5, 10 players on a team at a time are getting infected. I think this happened was it the Toronto team or somebody, maybe it was in Florida, I can't remember. And they basically had to shut down, the schedule for a few weeks, and while they resolved all this. So the golfing thing I think is the best analogy, and I think that could be a model for even schools, and for colleges,
But they're also the thing about golf though is you're outside and you don't have to stand
that's a good point.
People.
That's a good point. You can social distance. Yeah.
You've got a lot of distance. No one's really ever around you in the whole game,
Yeah.
Unless
that's totally true.
Have a caddy, So
yeah.
Just real quick. So we're getting to about 20 minutes here left.
Yeah.
Hey, we got the test. We had some notes here we wanted to find out about, which is are the with the kids and adults, like, are their symptoms the same? I've been hearing a lot about kids developing these weird other problems or symptoms. I mean, how do the kids and the adults relate in this?
Yeah. So it appears to be very different. And so very few kids, I mentioned in Florida, there's that 200 kids out of, I forget their number of cases is super high. Is it hundreds of thousands? What is it? But kids will be asymptomatic in general. And we won't even know, like they won't have a fever, nothing. There's a few reports, and this is where the disease gets a little crazy, of COVID toe. So what's really crazy is this virus is causing vascular problems, and it appears to like these cells called, I think they're called endothelial cells, which are like they line vessels basically. So what they're doing is causing damage to the circulatory system, the capillaries, and mainly in the lungs where many of those cells are. And then somehow they're getting into the bloodstream, and then they're going to places where they shouldn't. Like for some reason they're going to kids' toes, which makes no sense to me. But they get like these blisters, or like these it's not really blisters, but these red patches where there's clearly some sort of breach of the capillary system, the blood flow system in these areas. And so so that's the one thing I've heard about, but it's rare, and also the kids in general should be safe at large. But again, nobody wants, this is a rare, disease for kids, but we don't want to put our kids there. We'd rather, they spend an extra 6 months down the road, they're 18 and a half before they get out of, high school or whatever, it's like, we don't care about that. So that's the thing. And then for adults, I got another chart here too at this, ourworldindata.org. They show that where are the fatalities occurring? That's the thing. And so for kids, these 200 cases is fairly new. I don't know the details of that yet. But if we're healthy, you're going to be okay. Is the word. So if you don't have any, they talk about cardiovascular disease, diabetes, chronic respiratory disease like COPD, like from smoking, high blood pressure, cancer. The fatality rate is high, so it's like anywhere between 6 to 10% if you get COVID and you have one of those other diseases. If you have no health condition whatsoever, it's still too high for me. It's 1%. So if you get COVID, then there's a 1% chance you're not going to make it. And then that stratifies along age also. So that that's another metric in there. So so that's the difference. Is the older we get, and I think it's what's known as we get older, our immune systems become weaker. Like they kind of burn out. Like we've got these supercharged immune systems throughout most of our life, and then over time they start just getting weaker. They're not as good at doing their job. And I don't fully understand that. It's a genetic thing where I think that the genes don't translate properly. The cells lose their ability to recognize foreign invaders, and so that's the main thing that's going on there. And then if you have a health issue, then that system also the immune system I think is less mobile, I guess would be a word. So that's the big difference. Kids somehow are protected. Nobody understands that yet. As we get older, we're less protected, and then that's the danger. And we can have a whole bunch of symptoms like this thing's it's mainly a respiratory disease, which is where COVID comes from. Or SARS is a severe acute respiratory syndrome, but it can do other things. Like there's reports of it getting to the brain. There's these vascular things that can happen. It gets into the liver, to the kidneys, and so it's pretty crazy. It's in terms of morbidity.
Did you happen to find out? I mean, I know a while back, I just did a quick Google search, and I was like, finding out about the flu, and they said most flus originate China area, back in the deep country where all the farms are. I mean, can you tell us about that? Do you know anything about that?
A little bit. So I know
where these flus and SARS are. Where are they?
Where does it come from?
Where are they starting? And why?
So viruses are everywhere. They're all over the place. We have viruses that, like I studied a virus that we 8 out of 10 of us in this conversation have, and but you'll never know about it. It's like, so they're everywhere. And our immune systems, deal with them, or those viruses don't take off, and there's all kinds of reasons why it doesn't work. And so they're also in humans, and they're obviously, because that's when it becomes a problem. But they're also in animals. So they're in every mammal. So they're in mice, rats, squirrels, chickens, pigs, cows. They're everywhere. And so that's where it gets troublesome. And the theory for this particular virus is that it's really well known to infect bats. And for some reason, bats are carriers. Like they carry tons of viruses that people haven't even studied yet. And
oh, really?
And and then these bats are immune to the actual virus. But then if someone comes in contact with it, most of the time, I don't know, is it 900,000 out of 901,000? I don't know why I picked that 900,000, but it's highly improbable that virus will come and cause a problem for us because it's adapted to the bat or the mouse or whatever, and then we get it, but it doesn't recognize our biology, or our immune system clears it. But occasionally, the thing about viruses, they're constantly changing, and they're not completely stable, and this is evolution in a way. So the viruses are, it's not a single entity, it's constantly changing up its molecular structure a little bit, and that's where the strains idea comes from. So we talked about SARS-CoV-2, that's a particular DNA sequence, and then what can happen in mammals, and the reason that farms are important is because humans and mammals are in close proximity in many countries like China and elsewhere. And so there's this risk of what's called species jumping or species hopping. So normally the virus wouldn't cause a problem, but it can, with the right animal, one particular animal might have a slightly different version, and that species can hop. And that's what happens. And especially that happened with the flu, and I think with swine flu too, right? We heard of swine flu that jumped from a pig into humans. And there's what's called avian flu, which is from birds. So it goes from the birds to people. I even heard of one that started maybe in chickens, and it couldn't go to humans, but it went to pigs first, and then it changed into pig, and then it went to humans. So there's all this like hopping and natural selection that can happen.
Is there a traceability of these things?
It's really hard because you'd have to find animal zero, like the ground zero for that animal, and
so with the coronavirus, then is there the one we're dealing with? Is there an idea of like how it started?
Yeah, I keep hearing the suspicion is bats, and I think I heard pangolins early, but I think
we heard that early on. Yeah.
I think that was refuted, I think, and it wasn't there. And so this can be done, the studies can be done because there's tons of genetic information. We can like sequence anything like crazy now and get the whole genome of just about anything. And so those studies have been done, I think. And my understanding is it hasn't been pinpointed as of my last reading, which was probably about a month ago. Okay. So I don't think it's known yet, but there's, it clearly started in Wuhan, China, and then where did it come from? There's these conspiracy theories that it came out of a lab, which I highly doubt, or maybe really? It came out of some. Yeah, there's like a virology defense lab in Wuhan, and then there's a market there that they're claiming, and right. But I don't want to go too far on the virology lab. I've worked in virology labs and I've handled HIV, as I mentioned last time, and wow. Those people know what they're doing. There's all kinds of controls in place in China, and manufacturing and controls and environment. Like they've got all that. Like I just can't imagine that it just happened to get out, in a virology lab. So the most likely route, is that it was an animal hopping event, and that's what Fauci thinks too, and I think I've seen a few other people quote that as well.
Yeah, I think wasn't one of the early cases when it hit in Washington, we were one of the first ones, just like 2 hours south of here, and I think it wasn't someone who visited one of those live markets. So I mean that is possible, I just wondered if there's been any actual evidence of traceability back to either the lab or the market, if anyone knows for sure, but
not yet. That could be like a moment in time.
It's just like, hey,
it's curiosity. Yeah. How did this thing happen? It's a great question.
Yeah, or if I should get rid of my Wuhan gong that I have back here. So they do make really good trash cymbals and gongs. They have been for a long time. Neil Peart actually had Wuhan China symbol, and that's why I got one, is because he had one.
Nice.
And they sound incredible, and I have never heard one anything like it before.
Yeah.
But unfortunately,
we can still love Wuhan for their gongs.
I know, huh? But yeah, dang, man, that's crazy. There's a lot we don't know, and we're still trying to figure it out. Like today, we just went and got COVID tests in our nose today. So what test are they doing for that? Like, do you know? Like,
that's a PCR test.
PCR.
Yeah. So basically, and PCR stands for polymerase chain reaction. It's really cool. You basically the idea is if you have one, it's not as sensitive as what I'm saying, but like if you had one virus in your nose, they would capture that, and they actually capture probably, I think the sensitivity is like 10,000 copies is what they need, which is that's like, it sounds like a lot, but it's not that much. Right. And so they take the swab, and that's going to have a certain number of viruses on it, and maybe some viruses exploded and their DNA is just hanging out there. And so they put that in a test tube, and then they have these. The cool thing about DNA or RNA, they can do this for RNA too. They can amplify it, and so they use what are called primers, which are additional pieces of nucleic acid, and they're specific to that sequence. And then they basically take one copy or 10,000 copies and make make twice as many, and then so it's 1 goes to 2 goes to 4 goes to 8. That's the exponential thing, right? And then you do what are called cycles of amplification. So you basically recreate that gene, and then you recreate those 2 genes, and then you recreate the 4 genes, and then eventually you see a signal. And so that's super specific for the virus. And we talked about, is it, where do you get the test? I recommend going to your doctor, but what you probably got is probably relevant, most likely. And what you want to know is where did this test?
We went to the urgent care just down the street. So
yeah, that's going to be a real test.
Yeah. But I just wondered, how come they don't have these things like instantly, like you just put the swab in a machine and then it tells you?
Yeah,
I mean,
that's going to come.
That
will come, I guarantee it. Yeah, and it's called point of care diagnostics, and people are working on that. It's a little trickier because if you had a machine you could put a credit card in, right? You just put your stick in there, and it could.
Right.
I think that will come most likely. The machines need to be maintained, and you got to monitor their performance, every certain amount of time. And I could see where that could totally, happen, where we could go to some machine and get that done.
Wow. Okay.
Were there other points from last week that he had?
Yeah, Charles, did you have any? We got 5 minutes. Did you have anything from last week that you think or a couple of weeks ago when we last had you on, that you think is pretty important that people should hear?
I think, it's just minimize exposure right now. I think there's another thing that I kind of read is this transmission, right? I called it a super bug, which is kind of a description of how it can take off. And that could definitely happen. And I actually looked into that a little bit more. It's called transmission rates. And I thought it was a 2, like 1 person infects 2. It turns out it's not. It's actually closer to 1. There was an NPR report and some other people did it. Looked at it, and it's really close to 1, and so that's kind of good news. So it's not as bad as we think, but it is. You're welcome, Dan. Thanks for that.
Yeah, I see that.
So that's actually good news. And I think what's happened is maybe either there was inaccurate data in March is the last time I saw that number, or because we put controls in as a society, that we've dropped that from something higher than 1 down to 1.
Right.
So that's good news. But what we really want is for it to go less than 1, because that means people that are infected aren't giving it to other people. And so that's the thing. So we're not there yet. The masks can definitely help. They're not perfect. I saw somebody say that it's a 30% reduction in transmission, which doesn't sound like a lot, but it actually is, because of this slightly exponential. It's like 1.03 is the factor, I think, for transmission. So it's still important. So wear your mask, social distance. Don't go around people if you don't have to. But we got to work, we got to do our thing, we got to make money, we got to support our families, so we just got to balance it and just do the best you can where you are. I think that's the message.
Yeah, man. Well, dude, thank you so much again for coming
you're welcome.
on. I mean, this is amazing that you've been here at our disposal, and from somewhat of the inside of things, and just so everyone knows, if you're watching, this is Charles. He works for a well-known pharmaceutical company. He's 10 years in virology, so he knows what he's talking about. Yeah. And thank you again. And I'm sure we'll hit you up in a little while again, man.
Be happy to come back on. I'm watching it. It's totally fun.
What's up? You got any questions?
No, thank you, Charles. We appreciate it.
Yeah,
you're welcome.
Man.
Yeah.
All right.
Cool.
So let's finish up, and thank you again, Charles.
All right. See you all again.
All right, man. Thanks.
All right. You're welcome. Bye-bye.
Bye. All right. That's Charles Wartchow. Thank you again, Charles. He's awesome. So spread the word, you guys. We're doing these videos once a week right now. Herb's Happy Hour Fridays. Go to herbcider.com. We've got lots of really cool stuff.
Did you do an update on what's new and exciting upcoming for Herbs?
What's new and exciting, man? I kind of did. We got the pineapple margarita.
Birdie juice re-release. And it's
Birdie juice re-release. Lemon lime.
Available in stores next week.
And I think everyone can hear you.
Online, and the pineapple margarita, which we did a small batch. We're going to be releasing that in 6-pack cans, available online or for pickup at our production facility.
That's right.
Which we will be opening a new tasting room fairly soon.
At our production, we got our room almost ready.
We have an outdoor area there that is
you can have some space. You can wear a mask.
Well suited for COVID
drinking.
environment.
COVID drinking.
Yeah, and then we also have a new can label design coming out that's going to blow your mind.
I think I told
Did
them.
you guys talk about it?
I think
Okay.
we got some new can labels coming.
And then the bourbon barrel.
Yeah, I told them. We got bourbon barrel
those
coming
do
too. That's one of my
so
favorites.
The bourbon barrel that we're going to put into cans is actually going to have it's going to be co-blended with 3 different types of stone fruit. It's going to be a peach, apricot, and plum blend with the bourbon barrel in the can. So look for that
peach,
coming
apricot, and plum in the bourbon barrel coming out. Yeah, it's coming up very soon.
Herbcider.com for all your cider needs. Thanks, guys.
All right. Thank you guys very much. We're going to be ending here. Been reaching out to some people. We might have some cool people available coming up soon. So next week, Friday, stay tuned. Spread the word on the IG, Instagram, TV right here, Herb Cider. We've got a YouTube channel also. That helps spreading the word. And I think that's about it. You know, go on to herbcider.com. We'll ship the cans right to your door. You don't even have to get out and wear a mask or anything. You don't even need underwear. Just wait.
Good night. Happy weekend.
Yeah. Shama says good night. I say good night. Thank you for joining us, you guys.
Be safe.
And be safe. Wear a mask. Don't mess around.